Hi guys, Miss Francis here. Before we move on, I just want to take a second to review mitosis. You have these images in your note packet. Make sure you revisit them and can explain what's occurring during each stage during mitosis. Furthermore, if you're looking for another great animation, the McGraw-Hill Mitosis and Cytokinesis is an excellent resource. Stephanie Castle explains how you can determine the mitotic index. In order to calculate the mitotic index, you are given a micrograph picture where you have to count the total number of cells and then the number of cells that are in mitosis. You take the number of cells that are in mitosis, divide it by the total number of cells, multiply it by 100, and there you've got your mitotic index. Make sure that you are able to label all of the different phases depicted in these pictures. So if you want, you could pause, try and label them, and then check your answers. One is metaphase. 2 is anaphase. 3 would be interphase. 4 is prometaphase. 5 is prophase. 5 is telophase, and 6 is prophase. So make sure you'd be able to identify those. Furthermore, I gave you guys a couple of worksheets in class. This is the answer to the bottom of one of the worksheets that show these pictures where you guys have to label the different stages. Scientists hypothesize that mitosis in eukaryotes may have evolved from binary fission in bacteria. Now, binary fission and mitosis are sometimes confused because they're both types of asexual reproduction. However, there are slight differences. For example, mitosis involves phases or stages, however you want to call it, whereas binary fission is the simplest form of reproduction. There's no spindle involved, there's no sister chromatids involved, and there are no distinct cell cycle phases. So in binary fission, you've got a circular chromosome that replicates at the origin of replication site. Then those copied regions move to opposite ends, and scientists aren't really sure how that exactly occurs, what the mechanism behind that is. Lastly, the plasma membrane pinches inward to form 2 daughter cells, each with a complete genome. Scientists believe that there have been possible intermediate evolutionary steps between binary fission and mitosis, and these are explored in 2 different types of unicellular algae. So dinoflagellates are unicellular protists. 90% of marine plankton are dinoflagellates, and they could be photosynthetic or non-photosynthetic, where they get their food by feeding on protists. So in dinoflagellate, the replicated chromosomes are attached to the nuclear envelope, and then the next organism in diatoms, the spindle develops within the nucleus. Diatoms are photosynthetic algae, and these are just possible intermediate steps that occurred between binary fission and then the evolution of mitosis.